I am Gayatri, working as assistant professor in EC department at SNS College of Technology. Today I am going to discuss about the topic of enzymes, genes and chromosomes. First I am going to discuss about the DNA structures, which is nitrogenaceous base of DNA, purine base and pyrimidine base. The purine base, which is adenine and glycine, which is having the two carbon rings. Adenine, which is represented in A, and guanine, which is represented in G. And all the molecules, the purine bases and pyrimidine bases, will be having the carbon, hydrogen and nitrogen molecules, and oxygen will be present. And pyrimidine, which is having the thymine and cytosine. The thymine, which is represented in T, and cytosine, which is represented in C, which is having the carbon molecule, oxygen, and nitrogen molecule will be present in both. And the DNA will be having the chromosomes molecules, and gene will be having in the gene will be having exon, intron will be present in our gene molecules. The DNA chains, which is nucleoid, are joined with phosphodiester bond. The sequence of bases vary genetic information extremely will be having our long chain. The DNA represents the deoxyribonucleic acid, where it will be having both purine basis and pyrimidine basis structure, where it is having thymine, glycine, cytosine and adenine structure will be present in our DNA molecule. The DNA will be having the two polynucleotide chain, which is joined together, and the double helix twisted in right-handed way, full circle in every 10 bases. The DNA molecule will be in the form of ladder structure. The base will be steps, and sugar and phosphates will be supporting layers. The two nucleoid chain will be run in opposite direction and will be having our chemical reaction. The length of the DNA will be 34 and base pair spacing will be 3.4, and in between the supporting layer will be having sugars and phosphates. And then the DNA will be composed of chromosome, condensed cephaloid, fibers connected to chromosome cephaloid, chromatin fibers and chromatin. The chromatin, which is beads on the string form of chromatin, will be present, will be having the length of 11 nanometer, and the chromatin fibers, which is 30 nanometer chromatin fiber of packed nucleosomes, which is with having a distance of 30 nanometer. And fibers connected to the chromosome cephaloid, extended cephaloid associated form. The chromosome cephaloid will be present, and the diameter of the fibers will be 300 nanometer, and condensed cephaloid will be with the diameter of 700 nanometer, and the chromosome will be in the form of metaphase chromosome, with the diameter of with the length of 1400 nanometer. And gene will be having regulator elements, flanking region, exon 1, exon 2 will be present. The gene will be the DNA sequence that is needed to encode amino acid, the sequence of protein, composed of exon, intron and different control elements. All somatic cells, which is having 23 chromosome pair, 46 chromosome diploid cell 2N. The fertilization, N, male and female will be combined and produce a fertilized egg. The egg will be having 2N and 46 chromosome. The sperm cell will be having 23 chromosomes, haploid cell N, and the egg cell will be having 23 chromosome, which is also a haploid cell and have N cells. The chromosome pair will be having PP, AA and BB, genotype, homozygous for the dominant algae, homozygous for the regressive algae, and heterogeneous will be present. The local and algae body will be present. The meiosis, only in gamete formation, one diploid parent cell produce 4 haploid gametocytes. Mature gametocytes have 23 chromosomes, and the meiosis will be having meiotic division 1 and meiotic division 2 will be present. And crossing over, the chromatocytes change part between homologous chromatotides during the period causes the redistribution of hereditary material between chromosomes. The number of genes does not change. New alkyl combinations are formed during the crossing over. Thank you for watching.